All right. So we are technically recording right now. As you can see, there's a timer counting up from 0 at the top right.
Perfect. Yeah, got it.
So that'll help keep you on track. And then go ahead and click the next button for me. Now I should be able to see your slides moving. Yep, I see you on slide 2 now. It's perfect. Now slide 3. Okay, great. Everything's working as expected. Well, I will go ahead and end this little test session.
Hi, everyone. My name is Niren Murthy. I am a professor at the University of California at Berkeley in the Department of Bioengineering, and I'm also a member of the Innovative Genomics Institute. The title of my talk today is Lipid Nanoparticles with Acid-Degradable PEG Chains Efficiently Deliver Messenger RNA. As you all may know, lipid nanoparticles have great potential for delivering messenger RNA. However, before their vast therapeutic potential can be utilized, they need significant optimization. Lipid nanoparticles currently suffer from 3 problems. The first one is they are quite toxic. They're toxic for several reasons. One is they can potentially be immunogenic, and they also cause inflammation. We'll discuss some strategies to mitigate their toxicity at the end of today's lecture. The second problem affecting lipid nanoparticles is that they deliver messenger RNA very inefficiently. The vast majority of what they deliver gets trafficked to lysosomes and degraded there. And this prevents them from being used for a variety of therapeutic applications. And the final one, which I will not talk about today, but there are active projects in the lab on this, is lipid synthesis. Turns out that making lipids that are going to be part of lipid nanoparticles are actually quite challenging. And so people cannot make large libraries of lipids that can be used to screen lipid nanoparticles. And this is a pretty big roadblock in terms of lipid nanoparticle optimization. And from now, I'm going to use the acronym LNP for lipid nanoparticle. The first project I'm going to talk about today is a project that focuses on increasing the endosomal disruptive ability of LNPs. So as I said before, LNPs deliver mRNA very inefficiently because the vast majority of mRNA is trafficked to lysosomes where it is degraded. So there's great need in being able to enhance this endosomal disruption of LNPs. But before I get into our project, I'd like to discuss how it is that lipid nanoparticles actually disrupt endosomes. That's what's being shown on this slide here. Lipid nanoparticles contain an ionizable lipid. That's the yellow lipid on the lower left slide. It says ionizable lipid. The ionizable lipid is a lipid that has a pKa of around 7. So what that means is that at pH 7.4, it is by and large neutral. It's not positively charged because of the pH. And it turns out that at neutral pHs, and so when the ionizable lipid is neutral, so at pH 7.4, it's not charged, then it has a cylindrical shape and forms lipid bilayers. And because it's a bilayer forming lipid at pH 7.4, it's not membrane disruptive. This is absolutely critical. If it was membrane disruptive at pH 7.4, it'd be very toxic, too toxic to use. But then, once the lipid nanoparticle gets in the endosome, the ionizable lipid gets protonated. It then is hypothesized to flip into the endosomal membrane, which has lots of negatively charged lipids, in particular phosphatidylserine, and it ion pairs with the negatively charged lipids in the endosome. That ends up generating a lipid complex that has an inverted cone shape, and that disrupts the endosome. Now, for this mechanism to occur, as shown on the left, the LNP lipids have to be very close to the endosomal lipids. if they're not very, very close to each other, and when I say close, I mean 1 water molecule space or 1 nanometer apart, then the ionizable lipid cannot flip into the endosomal bilayer. Now, this is a big problem for lipid nanoparticles because lipid nanoparticles actually don't look like the yellow figure inside the endosome shown in the left of this figure. They look like this, the structure shown on the left. All lipid nanoparticles have a dense PEG chain. That PEG chain is shown on the structure on the left with the green dots on the inside. The squiggly lines coming off the lipid bilayer are the PEG chains. The PEG is this polymer that is essential for lipid nanoparticles. All lipid nanoparticles are now made with PEG chains. If you don't have the PEG chain, the lipid nanoparticles won't have the right size. They'll be too unstable, and they'll also be too toxic for use in humans. So you have to have the PEG. And the PEG works great for lowering toxicity and keeping the size low. But once your lipid nanoparticle gets in the cell and in the endosome, shown in the middle of this diagram, labeled endosome, then the PEG is a big problem because the PEG layer completely coats the lipid nanoparticle surface, and it prevents the lipid nanoparticle surface from getting close to the endosomal bilayer. So you can't get the flipping of the ionizable lipid into the endosome. And so that is one of the reasons why LNPs are so inefficient in endosomal disruption. Well, a potential solution to this is to simply make lipid nanoparticles where the PEG chains come off under acidic conditions. That's what's shown on the right of this figure. If you were able to do that, then you would have a surface of your LNP that's completely unhindered and can come really close to the LNP, to the endosomal surface, and allow the lipid flip-flopping to occur, and then thus endosomal disruption. People have been trying to do this for about 30 years. It's actually quite challenging. And this is because the endosomal pH is only about pH 6. So not a big change from the pH 7.4 environment of the blood. In addition, endosomes get trafficked to lysosomes on the timescale of 30 minutes to 60 minutes. And in the lysosomes, they're almost degraded instantaneously. So it's quite challenging to make a linker that's for making a PEG conjugate of a lipid that hydrolyzes on the timescale of 30 to 60 minutes at pH 6, but is stable enough to do synthesis and formulate LNPs. To address this challenge, our lab developed a new class of linkers shown here called the azide acetal. The azide acetal is the blue structure shown in the dotted lines. So it's an acetal that has an azide in the para position. This is a unique linker because its half-life at pH 7.4 is 19 days. It's quite stable. But if you now, and then the idea is that you can formulate LNPs with it, and once you've made these LNPs, then you mix your LNPs with DTT. DTT reduces that azide to an amine, and now it has a half-life of 15 minutes at pH 6.0, which is the endosomal pH. So we've now managed to make a linker that's very, very stable enough to do multi-step synthesis and also formulate LNPs, but also rapidly hydrolyzes in endosomes. We've made a lot of molecules with this linker. The one I'm going to talk about for the next few slides is a molecule shown on this slide that has cholesterol on one end. That's the reddish or pink structure. And then PEG chain on the left side. That's the green structure. The rationale for making this acid-degradable PEG lipid is shown here. That's we call these acid-degradable PEG lipids ADPs, or the number 1, and that's shown on the left of this slide. And so the rationale is that if you make LNPs with the ADPs, you get LNPs as you normally would, but once these LNPs get into endosomes, which is shown on the upper right, then the PEG chains come off and the LNP can now fuse with the endosomal bilayer and allow the lipid flipping to occur, and then endosomal disruption and your mRNA comes out. In contrast, traditional LNPs shown on the lower part of this figure form LNPs beautifully, but the PEG chains do not come off and you're sequestered in endosomes. One nice thing or one interesting thing about the ADP LNPs is that they tolerate very high levels of pegylation. We've made a lot of different ADP LNP formulations. The one that works the best are the ADP LNPs that have 10% PEG. That's about 10 times the amount of PEG on a regular LNP, which have about 0.5 to 1% PEG. It turns out that having large amounts of PEG can be beneficial for multiple reasons. It enhances the circulation half-life, and it also enhances the diffusion through dense tissue. And this can be very helpful if you're trying to transfect tissues like brain where you have to do an intracranial injection. I'll discuss some data on that later today. We have investigated the ADP LNPs in multiple contexts. One experiment we did was on human hematopoietic stem cells in vitro. Transfecting human hematopoietic stem cells has numerous applications because hematopoietic stem cells are the material used to generate all types of cellular-based therapies, from bone marrow transplants to a variety of others. But transfecting hematopoietic stem cells is actually quite challenging. Right now, it's done via electroporation. That turns out to be exceedingly expensive to do in a clinical fashion because of the manufacturing costs. And there's great interest in developing alternatives. But lipid nanoparticles do not generally transfect hematopoietic stem cells because of their high toxicity. We investigated if having a 10% PEG instead of 1% PEG could solve that problem. So we investigated if ADP LNPs can deliver GFP mRNA to HSPCs. That's the graph in the middle. The green bars is cell viability, and the gray bars is the percent transfection. So you can see here that electroporation will transfect about 80% of the cells, but it does cause a lot of toxicity. Your viability is only around 30%, so 70% tox. That's also quite bad because that also means that you're changing the phenotype of the remaining 30%, most likely. But if you deliver with ADP LNPs, your transfection efficiency is slightly higher than what it is for electroporation. It's close to 90%. And your viability is also higher. It's about 60%. So we think that ADP LNPs could have some applications as cell therapy reagents. ADP LNPs were also able to deliver Cas9 mRNA and guide RNA and edit HSPCs. There are numerous applications where mRNA delivery can be potentially therapeutic. Of these, the brain is one of the most exciting, in particular in the context of gene editing. And this is because most brain diseases, such as Alzheimer's, Parkinson's, traumatic brain injury, are untreatable. And in general, it's very hard to develop therapeutics for brain diseases because of the blood-brain barrier. Gene editing administered via an intracranial injection of gene editing enzymes encapsulated in LNPs has great potential, we think, because you'd only have to do it once because gene editing is permanent. However, despite its tremendous potential, very little is known about gene editing in the brain. So we started a collaboration with Professor Hae-Young Lee at the University of Texas in San Antonio to investigate if ADP LNPs could transfect brain tissue after an intracranial injection. The first set of experiments her lab did was using the AI9 mouse model. This is a mouse model where if you deliver Cre mRNA, the cell turns red permanently. So we made ADP LNPs with Cre mRNA, we shipped it to her lab in Texas, and she injected it into the hippocampus. And she then counted the number of red cells in the hippocampus. The results are shown here on the left. The y-axis is the percent TD tomato positive cells. The x-axis is the various groups. And what you can see here is that at a 2x dose of Cre mRNA, we transfected about 30% of the cells in the entire hippocampus after a single intracranial injection. That's actually quite promising because at that level, you can probably treat a lot of different diseases where hippocampal damage is a major issue. Based on that, we also investigated if ADP LNPs could deliver Cas9 mRNA and guide RNA to brain tissue. And so we made LNPs with Cas9 mRNA and guide RNA targeting the AI9 locus, shipped it to Hae-Young's lab. She injected it into the hippocampus. And compared them against standard LNPs with just 1% PEG. So the hypothesis underlying this experiment was that the dense pegylation on the ADP LNPs should enable them to diffuse through brain tissue better than just 1% PEG. And the results are shown here on the left. And so you can see that ADP LNPs transfected a much larger number of cells than standard LNPs in the brain after a direct intracranial injection. In fact, the number is about 5 times higher. And we're now investigating this in various disease models of the brain. On the next few slides, I'm going to talk about another collaborative project using ADP LNPs, and that's for doing gene editing in the brain in utero. And this is a collaboration with Professor Aijun Wang at the University of California at Davis. In utero gene editing has tremendous clinical potential. And this is because there are numerous genetic diseases of the brain, like Fragile X syndrome. These diseases are devastating. There's no cure for them. And in a lot of cases, the damage to the baby is done by the time the baby is born. So if you wait until the baby is born to try and correct these diseases, it's too late, because once the brain is deformed or has developed improperly, it's impossible to actually rescue, or at least very, very difficult. However, a lot of these genetic diseases could potentially be corrected by delivering gene editing enzymes in utero. In addition to this, the in utero environment is very likely to be highly permissive to gene editing. And this is because the blood-brain barrier has not been formed. The immune system also hasn't been formed, so you don't have to worry about the toxicology. The number of cells is very small. And finally, and perhaps most importantly, the number of stem cells is also very, very large. So you have an opportunity now to transfect a large percentage of the total number of cells in a person by a simple intracranial injection. However, despite its promise, very little is known about in utero gene editing of the brain. So we did an experiment with Professor Wang's lab where we shipped them LNPs, ADP LNPs that contained Cre. His lab injected them into the ventricular space, so an ICV or intracerebroventricular injection in utero. And this is done in the fetus of AI9 mice, and we then looked to see what the toxicity of these LNPs was, compared them to standard LNPs, and then we also did a series of investigations to determine what cell types were transfected and how efficient it was. So the first thing we did was look at the toxicology of these LNPs. And that's shown on the left. And so what you can see here is that the ADP LNPs had pretty well tolerated. So they injected 14 fetuses, and 12 out of the 14 fetuses were born and survived well. And that's about what you would see from just PBS. It turns out that doing the ICV injection in utero, there's a certain amount of toxicity just associated with that, and we're not increasing that with ADP LNPs. But in contrast, if you use standard LNPs, shown on the left here, called LC3 LNPs, those are the standard LNPs with 1% PEG, 18 out of the 19 fetuses died. So only 1 out of 19 fetuses survived versus 12 out of 14 fetuses surviving in the ADP LNP case. So we think that ADP LNPs, because of their dense pegylation, are actually less toxic than traditional LNPs. In addition, we also investigated what the transfection efficiency was of the ADP LNPs in the brain. And the way Aijun's lab did this is that they injected ADP LNPs with Cre mRNA in utero, ICV, and then after 48 hours, they sacrificed the fetus and did flow cytometry on the brain. The results are shown in the middle in the bar graph, where the y-axis says percent TD to medial positive cells. And what you can see is that we transfected 30% of the total number of cells in the brains of these fetus. That's just a remarkably high number. There is no way to achieve that transfection rate in an adult. In fact, if you were to do this experiment in an adult mouse, you would transfect less than 1% of the total brain cells. probably like 0.3% or 0.1%, something like that. So this is orders of magnitude higher. And it's actually quite exciting. In addition, we also saw that we were transfecting the spinal cord. Again, it's very, very hard to transfect the spinal cord. In the figure in the middle on the lower, with a panel of 3 figures, you can see the long, skinny red streak, and that is the spinal cord being transfected. So we got pretty good efficiency in the spinal cord. In addition, we also look to see what happens at 10 weeks of age. So we let these mice, we did a separate set of experiments where we did an ADP LNP injection in utero, and then we let the mice be born and waited 10 weeks. And then we isolated the cortex and the hippocampus and did flow cytometry on them to see what was the percentage of cells that were transfected. Now, the thing about the Cree model is that every time, if a cell gets Cree mRNA in utero and becomes red, that red gene is now propagated to the daughter cells. So when those cells divide, their daughter cells are also red. So that's why we can do this experiment at 10 weeks of age. So these are almost adult mice. And the result here is on the right. And I apologize for some of the slides not showing up here, but the key slide is actually the key data is actually on the right, where it says TD tomato positive cells, cortex, hippocampus. So what this figure is showing is the total percentage of cells in the cortex and hippocampus that were transfected at 10 weeks of age. And we transfected 40% of the cortex and 60% of the hippocampus. Again, it's just absolutely impossible to come even close to this if you were to try and do an intracranial injection in the cortex or hippocampus of a mouse that was actually 10 weeks old. You have to do it in utero if you want to get these numbers. And with these numbers, you can really do a lot of therapeutics. You can also really, I think, treat a lot of diseases. Finally, we investigated if we could deliver a therapeutic guide RNA with ADP LNPs in utero. We selected the Angelman's disease for an initial test of the ADP LNPs. Angelman's is a devastating neurological disease caused by mutations in the UBE3A gene that basically dramatically lowers it. Turns out that this only happens in one allele. And that in patients who have Angelman's, they have another allele that actually has functional UBE3A, but it's being inhibited by something called the UBE3A ATS, which is an antisense that is made by that allele that shuts down the UBE3A gene. So this is remarkable because it means that there's potential therapeutic for this disease. All you have to do is knock out the UBE3A ATS, because that's producing an antisense that shuts down the UBE3A in the functional allele. So we made, and people had been investigating this for a long time. There are guide RNAs that have been already designed that can do this. We took those guide RNAs and Cas9 mRNA, packaged them into the ADP LNPs, injected them in utero ICV into a wild-type mouse, and investigated if we could generate indels in the UBE ATS. The results are shown here. And in this experiment, we sacrificed the mice 10 days after birth and did Sanger sequencing. And under the graph labeled B, the y-axis is percent indels, and you can see that we edited 20% of the UBE3A ATS gene in the entire brain at 10 days after birth. In addition, we were also able to downregulate the amount of UBE3A ATS that's shown in C. And so we got about a 30 to 40% reduction, which was statistically significant. We're now going to move on to disease models. So this was done in a wild type mice. We're now going to start working on the disease model mouse. Okay, in the interest of time, I'm going to switch gears and talk about a project in the lab that focuses on lowering the toxicity of LNPs. And the one thing we're going to focus on is trying to lower the toxicity of the PEG chain. Now, it turns out that PEG has many beneficial properties and is absolutely essential for LNPs, but they do have one problem. and that is that they are a foreign polymer, and because they're a foreign polymer, PEG can sometimes cause antibodies. That's what's depicted here on this slide here. It's been widely known that a lot of PEGylated therapeutics, after the first dose, will cause the production of IgM and IgG, and that can be very detrimental because when you do the second dose, this IgG will start binding the PEG on your therapeutic, and that will cause the activation of complement, which will cause tremendous amounts of inflammation and will also lead to phagocytosis and the elimination of your product. And the significance of this PEG problem was not fully recognized until recently. But now that everybody has, or at least 1 billion people in the world have received the Pfizer and Moderna vaccine, it turns out that we all have been immunized against PEG. And this is because both the Pfizer and the Moderna vaccine have PEG on them. Those are PEGylated LNPs, like all LNPs. And so this has actually now been measured. And that's what I'm showing you on this slide here. So the y-axis is the amount of anti-PEG IgG in patients that have gotten the Pfizer and Moderna vaccine. And if you look at D, that's probably the most understandable figure. That's the log change in IgG in patients who received the Pfizer vaccine, which is the BNT, or the Moderna vaccine, that's the mRNA 1273. And they're being compared to unvaccinated. And the take-home message from this is that if you've been vaccinated with the COVID vaccine, you have PEG antibodies. Your PEG antibodies are about 10 to 15 times higher than what they were in unvaccinated people. And this is 6 months after the second dose. And most interestingly, the amount of PEG antibody increases with every dose. So this, we think, is going to be a very big problem in the future because it could cause devastating consequences for people who receive intravenous injection of LNPs. Most future-based LNP therapies are going to be IV injected. So there's a great need for developing alternatives to the PEG. People are working on this pretty intensely, and two options have already been discovered: polysarcosine and poly-2-ethyloxazoline. Both of these can replace the PEG on LNPs. They make LNPs that function perfectly well, but they're also foreign polymers, and they also themselves generate PEG antibodies. And they have some biocompatibility issues, so it's not clear if they're really going to be suitable PEG replacements. Too early to tell. So other alternatives are also needed. One polymer that we think is quite interesting is a polymer called PGE. And that's the chemical structure is shown here. It's a polyglutamic acid, which has been esterified with short ethylene oxide grafts. The rationale for this, for having this type of a polymer, is that it turns out that PEG antibodies only bind PEGs of 7 units in length or larger. They can't bind short PEG fragments. So what that means is, and what this has also been demonstrated, which is that instead of having one long PEG polymer like you now do on LNPs, if you were just break that PEG up into small little fragments and graft them onto a polymer backbone, that generally does the same thing as a long linear PEG, but it doesn't induce the antibodies because you never have a long enough fragment to bind the antibody. So that concept had been out of the literature for a long time, but nobody ever tried to use PGE for making LNPs. We were able to do that by conjugating it to cholesterol, shown on the synthetic route here. And we then made LNPs with it. In interest of time, I'm going to move a little bit faster. And we made LNPs with it, with PGE LNPs, and injected them into the liver after an IV injection. And we injected them via IV and measured the amount of liver transfection using luciferase mRNA. The results are shown here. And what we can learn from this is that the PGE LNPs work about as good as the PEG LNPs. So based on this, we then investigated, can you do chronic dosing of PGE LNPs because you won't form PEG antibodies? And we did that using EPO mRNA as the model system. The results are shown here. The y-axis is EPO concentration, and the x-axis is the number of doses The green bar is the PGE-LNPs. And as you can see, after the third dose, the PGE-LNPs are much, much better than the PEG-LNPs. And this is presumably because of a lack of antibodies. So we're now looking at this for various types of chronic diseases. So in conclusion, I've talked about the ADP LNPs, acid-degradable PEG LNPs. They transfected hepatocytes and brain tissue very well. The PGE LNPs were able to replace the PEG on LNPs. I do have some conflict of interest. I'm a co-founder of the companies GenEdit, Opus, and Microbial Medical. And I also, before I end, I just want to thank the people that did the work. The ADP LNPs were done by two very talented postdocs, Hisong Han and Sheng Zhao. And the PGE LNPs were done by another talented postdoc, Yaolin Li. I want to thank the funding agencies for their funding. And finally, I'd like to thank you for your attention. And if you have questions, please send me an email.